Welcome everyone, and thank you for joining us for this live panel discussion. My name is Dr. Donna Reece, and I'm pleased to have with me other guest members from CMRG and our international consultant. And we will be reviewing and discussing some of the highlights from the 62nd annual ASH meeting. This is our first CMRG ASH review, and we're delighted to have a chance to present these updates in this new format. Before we start, I'd like each of the panelists to briefly introduce themselves.
My name is Arleigh McCurdy. I'm a hematologist and myeloma specialist at the Ottawa Hospital and the University of Ottawa in Ottawa, Canada.
I'm Darrell White. I'm a hematologist in Halifax at the QE2 Health Sciences Center in Dalhousie University.
I'm Dr. Richard Leblanc. I'm a hematologist and a medical oncologist in Montreal, working at Hôpital Maisonneuve-Rosemont. I'm a specialist of myeloma.
I'm Julie Stakiw. I'm a hematologist with the Saskatchewan Cancer Agency in Saskatoon and the University of Saskatchewan.
Hi, my name is Ajai Chari. I'm a professor of medicine and the director of clinical research at the Mount Sinai Myeloma Medical Center in New York. And it's a pleasure to be the honorary Canadian today.
Thank you, everyone. We have quite a busy agenda, and we have 20 oral sessions that we'd like to review. The discussion session will be followed by a question period at the end, and we do invite you to submit your questions using the YouTube live chat throughout the session, and we'll address as many of these as possible at the end. The first portion of the discussion will cover these oral sessions, including clinical data beginning with refractory myeloma, and then moving on to the management of newly diagnosed disease. Without any further delay, I'd like to present our first panelist, Dr. Chari, who will discuss abstract number 131 as we begin the section that will talk about CAR T cells in multiple myeloma. Ajai.
Hi. So we know that the Bluebird has already been published in New England, and the question being asked in this abstract is, how can we do better? And there's thought that if we have memory T cells that linger, that might increase the persistence of the CAR. So here, the BB21217 adds a PI3 kinase inhibitor to try to increase the presence of memory T cells. And what they demonstrated is that the response rate overall was 60%, but with this undisclosed new manufacturing method, they were able to demonstrate in a smaller group an 84% response rate. We don't have PFS, but the median duration of response was very encouraging at 17 months. And that extra response didn't seem to come with any additive toxicity. So I think the take-home message, what we always struggle with CAR T, it's that whole risk-benefit, especially in Canada, cost. The risk here, we know is manageable CRS, but there are some infections and cytopenias. The benefit is very compelling initially, but now I think the bispecifics are really giving them a good run for their money. And when we think about the tremendous patient selection going into CAR T in terms of excluding explosive disease, the manufacturing period which eliminates additional patients, and then finally getting to that actual infusion, if we did an intention to treat analysis, we might do quite well, I think, even with bispecific. And without that plateau that we're seeing in other hemalignancies, the question is really going to be, will the cost of CAR T be justified in this very, very competitive space? So, but very interesting preliminary data about this, the addition of PI3 kinase and the correlatives with the memory T cells.
Thank you, Ajai. I think this represents a second generation, if you will, of CAR T, trying to build upon the initial experience in myeloma. And Dr. McCurdy, if you would please provide an update of abstract number 131, which is a review of the ide-cel, the BB2121, before the modifications that Dr. Chari just presented. That gives us a bit of a baseline to compare a number of immune platforms.
Thanks, Donna and Ajai. So yeah, the ide-cel study that we're looking at really focused on BB2121, specifically the dose escalation and dose expansion cohorts. And so this was looking at patients who were heavily pre-treated. Most of the patients were triple exposed and triple refractory. And we saw some very impressive overall response rates. So about 75% in a heavily pre-treated population, which is excellent. The toxicity seemed manageable, and that's very encouraging as we move forward with these products in Canada. I think, you know, one of the concerns that has been raised is just where is this going to go in the next few years in Canada? And I think the cost barrier remains. This is a very expensive therapy. We have some geographical issues to work on in terms of getting this widely distributed. And again, this is a competitive space with the bispecifics and other agents coming through. I'm really encouraged to see the positive results in this study, but I am unsure as to where this is going to land in the next few years.
Thank you. I think we're all watching this closely, and I appreciate this overview. I think two of the issues we've all noticed from the CAR T-cell experience is the selection bias, who actually gets there, the dropout rate, both after collection, because there's a quite long lag before the cells are actually infused, and patients may deteriorate and not be able to get their CAR T. And also a challenge is the durability of these CAR T cells. So I think we'll switch gears a bit in the relapse setting and talk about some of the studies that use agents that are available to us now given in different ways, particularly the CD38 antibodies. So I wondered if Dr. Leblanc could give an update of abstract 414, which uses isatuximab in this setting.
So the IKEMA trial is a phase 3 trial that compared isatuximab carfilzomib dex versus carfilzomib dex alone, and it has already been presented on a previous meeting. Here, the question of this presentation is the addition of isatuximab to carfilzomib and dex increase depth of response and response kinetic in comparison to carfilzomib and dex alone? And what they found is that obviously for what we expect is the addition of isatuximab certainly increased the response rate, but modestly, but mostly the quality of response, much more very good partial response or better complete response rate, and probably most importantly, higher MRD negativity results for patients receiving the addition of isatuximab. So there are some limitations to this trial since today, most patients have already received anti-CD38 monoclonal antibody at the time we are using carfilzomib in treatment. However, for the few patients who have not been exposed to this category of medication, certainly IKEMA is a practice-changing study since probably the addition of isatuximab to carfilzomib and dexamethasone is much more attractive than our standard practice actually with carfilzomib used with dex or KCD, for example.
Thank you. I think we will continue this line of studies by talking about another antibody combination in the Apollo trial. This one does not utilize carfilzomib, but Dr. Chari, would you update us on the Apollo trial, which is abstract number 412?
Yeah, and I think it's an interesting contradistinction to what Dr. Leblanc just presented, because here we have the POM-DEX backbone with the addition of sub-Q DARA. In the US, we already had DARA POM-DEX approved on the basis of a phase 1 study with 100 patients, but here's the confirmatory phase 3, which most countries, including Canada, need for approval. And it's POM-DEX versus the addition of CD38 DARA. And I think as expected with many of these CD38s, we did see a benefit. And so we saw in the control arm a PFS of 6.9 and the addition of DARA was 12.4. with 37% improvement. And so that's, I think, encouraging. And but when we drill down to Len refractory, which is an increasing issue in patients who are getting frontline therapy, that PFS was 9.9 months versus 6.5, hazard ratio 0.36. So again, if you look at the hazard ratio, that seems reasonable. But to me, and I should just mention that there was no real unusual safety signal. We did see a slight increase in neutropenia and slight increase in infections. But overall, the PFS benefit was there. But to me, the main question that we need to think about is we now have 2 studies with POMDEX backbone, Apollo and Icaria, and then we have 2 with KD backbones, Candor and Ikema. And they all 4 seem to give the similar hazard ratios, but the key question is, what is your control arm? And so here we have a homeopathic POMDEX backbone, and of course you can do better, but the Ikema and Candor should be commended on using not just a homeopathic KD at 27, but they were using 56 twice weekly, which is Endeavor dosing. So if you could improve with a hazard ratio on top of a really viable option, I think it's showing the value added. And to me, these 4 studies, to me, is really practice changing, because I think it tells us the importance of class switching. And so if you have Len refractory patient, we do see the benefit of KD and the addition of CD38. But of course, I will leave the discussion of cost to my Canadian colleagues, because that is very important as well.
So that is a great lead-in and a great overview, Ajai. I'd like to call on Arleigh again to present the Canadian experience, which uses a POMDEX backbone, but with a twist that takes into account cost and hopefully can address this almost unmet need, or better phrased, ill-defined need of what we do for patients progressing on lenalidomide first line, which, as you pointed out, is increasingly the type of patient we're seeing in clinic at the time of first relapse.
Thanks, Donna. And so we'll see my Canadian showing a little bit with talking about this study. So this is our CMRG trial 004, which was a randomized phase 2 study comparing 2 arms. Arm A was a quadruplet with a combination of daratumumab, cyclophosphamide, dexamethasone, and pomalidomide. And arm B being daratumumab, cyclophosphamide, and dexamethasone with the pomalidomide added at progression. And we've talked a little bit about how Cytoxan is sort of a go-to for us, and many of us have had lots of mileage from that in the clinic. And of course, there's some immunogenic effects that make it appealing to study as well. And so I should mention that a lot of the correlative biology of this trial, the studies are ongoing. This was a study of relatively heavily pre-treated patients. So it was 120 patients. Most of the patients, in fact, 96% of them were lenalidomide exposed, and 2/3 were lenalidomide refractory. And when we look at the overall response rate, it was impressive, particularly in the quadruplet. It was over 88%, whereas it was in the 51% in the group that had the daratumumab, Cytoxan, and Dex. However, most of those patients were able to recapture response when the pomalidomide was added. I should also mention that the PFS, despite a follow-up of 25 months, was not met in the quadruplet arm A, and is projected to be around 30 months, median 2 prior lines of therapy. And in fact, when looking specifically at the lenalidomide refractory population, the PFS was 26.9 months, which is certainly better than what we've seen in the other POM monoclonal antibody combinations with a seemingly similar patient population. So, you know, I think with a relatively, I called it cost neutral intervention of cyclophosphamide the other day, we're seeing some of these patients. And I think it begs the question of what is our go-to treatment in patients progressing on lenalidomide who have not had exposure to a monoclonal antibody or an anti-CD38 monoclonal antibody. And Candor and Ikema, and in my mind, this cocktail, including the cyclophosphamide, are all contenders in that space.
If I may just add, I was actually an abstract reviewer this year, and I really loved this abstract, and I think it's our first, and maybe I'm showing my honorary Canadianness, but I really liked, you know, first of all, it's the first quad that we've seen in relapsed myeloma, and it's a randomized study, and I think it's cost effective, and I think you hit it on the head, which is it's really now CD38 KD versus this quad, to me, is the main choice, because I think not including the cyclophosphamide is really not good enough. Who's happy with a 9.9-month PFS for a LEN refractory patient? So I think we need to do better, and I think I commend you all for doing this work, and kudos, and I think this will be practice changing as well.
Thank you. We were excited to do this, and I think there are ways this regimen could probably be tweaked to make it even a little more user-friendly. Arleigh, would you like to comment a little bit on the toxicity or the practicalities of giving this quadruplet?
Yeah, so it's a good point and a good question. You know, I think it's fair to say that there was significant cytopenias, particularly neutropenia, seen with this, particularly arm A of the study, and growth factor support was needed in many patients and used in many patients. I think there are different ways you could finesse the dosing of both the POM and the Cytoxan. I would love to see this using metronomic cyclophosphamide as well, which may be a gentler option on the counts. So I found it manageable with growth factor support.
Yeah, I think it's probably beyond the spectrum of this discussion, but we would love to modify the quadruplet arm a bit by giving cyclophosphamide, as you said, at a lower dose, maybe 50 milligrams every other day. We do not have data for that, but would like to explore it in the future. But I think cyclophosphamide has been a workhorse for us, both with CyBOR-D and now with this quadruplet. I would like to point out that even the triplet arm did better than one would expect in terms of getting a decent progression-free survival, which might be of interest to jurisdictions that don't have access or funding for pomalidomide. So thank you for that discussion. I think we will now turn from the agents we have available to some of the newer antibody studies that include the bispecific T-cell engagers and conjugated antibodies. So I'd like to ask Dr. Darrell White to give us an overview of abstract 181, which talks about the Amgen bispecific, or BITE as it's nicknamed, in relapse refractory myeloma.
Thanks, Donna. So this trial was a first-in-human presentation of AMG 701, which is built on the AMG 420 drug, which was shown to be effective in heavily pretreated patients, with the major drawback being some of the safety issues, but more so the inconvenience of a continuous infusion. So the drug worked, but it had to be infused continuously. And the major benefit of the AMG 701 is that it can be infused intermittently. So both drugs are anti-BCMA antibodies, which engage T cells through CD3. This drug was trialed in this case in a dose escalation manner in order to establish safety and tolerability and the active dose level. So starting out at very low doses and building up. The patients included, so there are 85 patients included, most were treated at relatively low levels of drug. All were heavily pretreated. Median number of prior lines was 6. And about a quarter of the patients additionally had extramedullary disease at the time of inclusion. They did begin to see some cytokine release syndrome at a relatively low dose, but even at the higher doses, the cytokine release syndrome, as it became more common, was primarily grade 1-2, with a small percentage, I think less than 10% of grade 3, and no grade 4. They did see hematologic toxicity commonly. Non-hematologic toxicity was mostly GI, fatigue, and fever, so quite manageable. And there were 4 deaths on this study, 2 from sepsis that was said to be unrelated, and 2 from bleeding, one being a retroperitoneal bleed and one being a subdural hematoma. Overall, for the 85 patients, response rate was about 25%. And in those treated with at least 3 milligrams, it was 36%. And in those treated with the 2 highest doses, which really was only 6 patients, there were mainly responders, I believe 5 out of 6 patients responded, including some patients achieving MRD negativity, which is, I think, remarkable with that heavily pretreated group. And they did PK work as well that supported the weekly dosing, which is encouraging. So I think this trial won't have any immediate effect in terms of our practice, but will lead to further study, and the drug is still promising, and I think we'll probably see further study with it.
Thank you. This is a ways from coming on the market, of course, or even being fully studied, but there are some centers in Canada that are participating in this trial. And now that we're at a dose where 5 out of 6 patients have responded with very advanced diseases, I think there's a lot of hopes being pinned on this. And as has been said earlier, the bispecifics such as this, we feel are going to give CAR T cells a run for their money, if you will. The other type of antibody that theoretically offers advantages over the CD38 ones on the market, daratumumab and isatuximab, are the so-called conjugated antibodies, in which an antibody directed against a myeloma cell antigen is attached to a toxin. So you get not only the immune effect to kill the cell, but you also deliver a toxin directly to the cancer cell. So I will ask Dr. Stakiw to please present the experience with belamaf, one of these conjugated antibodies. This is abstract 725.
This is another CMRG study, the Algonquin study led by Dr. Trudell, which looked at belamaf, a BCMA drug conjugate with pomalidomide and dexamethasone. And really this part of the study was trying to find the ultimate dose of belamaf and the way to administer it with pomalidomide and dexamethasone. There are 4 different arms for this study, and 35 patients were presented by Dr. Trudell at ASH. Essentially, while there was a great VGPR or better found with belamaf and pomalidomide and dexamethasone, particularly in the 2.5 milligram per kilogram dose, I think Dr. Trudell emphasized that the keratopathy is going to be the rate limiting step in this study going any further right now. Almost 50% of patients experience keratopathy, whether that was from an ophthalmologist finding or whether those were symptomatic patients. But she indicated during her presentation that right now they're trying to find the optimal administration of this dose, whether that's every 8 or 12 weeks, that makes it a little bit more tolerant for patients with the pomalidomide and dexamethasone backbone. So still not ready yet for prime time in Canada. We have access compassionately, which has been nice because it's been great for us to get some experience with the drug. And it'll be exciting to see where this goes.
I think it is a very competitive field. We've only scratched the surface of these different antibodies that are coming along fast and furious in different trials. And we focused on those that have some relevance or availability via clinical trial in some centers in Canada. And we might add that the belamaf is available through a special access program through Health Canada on an individual request basis. So stay tuned for more updates while this trial continues to find that balance between toxicity and efficacy.
Can I just add one point to this? I know we're just, I don't want to delay things too much, but I just think with this study, the keratopathy does need work, and we need to figure out the dosing schedule, but the drug works, and it works quickly, and it works in almost all patients. And so I think despite the challenges that we have to figure this out, I do think that this drug has potential in myeloma, and so I'm quite excited about it, actually.
Thank you. Any other comments about belamaf or any of the antibodies that were discussed at ASH that we've not presented in detail?
I would just like to add, I think if we zoom out at the whole kind of spectrum of what's available in the antibody space, I think Actually, if you're interested, there's an education session where we summarize all of these data for you. And so we covered, I think there's now 7 BCMA bispecific antibodies. And I think as a group, at therapeutic doses, they're all demonstrating response rates on the order of 60 to 80%, which when you think about other drugs that in this same space have gotten what we call accelerated approval in the US, that's Dara, Carf, Pom, Belamaf, and selinexor, typically we get 20 to 30% for 3 to 4 months. And here we're seeing 60 to 80% in patients who failed a lot of those drugs for very durable. And so I think this is really, cannot state how exciting this is. And I'm really excited to see these move up. I think the ADCs have going for them the convenience of being given in the community, but I struggle, I think, as has been said, with the toxicity, because, you know, for a community doctor, we need to give a clean, simple schedule. No, do this, do that, play this. What is the dose and schedule and combination? And that needs to be really straightened out. And then I should mention there's also the MEDI2228 data that was presented, which is another ADC against BCMA, which did not have any keratopathy because its conjugate is PBD or pyrrolobenzodiazepine. But there we saw some dry eyes, pleural effusion, and rash. So each drug comes with its own baggage, but I think lots of choices, which is great for patients.
Thank you. I think that pulls the topic together beautifully. And we're going to change emphasis a bit from the immune treatment right now and talk about some new molecules that are more familiar in their mechanisms and their route of administration than the immune platforms like CAR-T or the bispecifics. And I'd like to begin by asking Dr. McCurdy to talk about the new CELMoD combinations using iberdomide. This is abstract number 724.
Sorry, unmute myself. Thanks, Donna. So this study was actually a couple of arms of a backbone study. So this was looking at the maximum tolerated dose and recommended phase 2 dose for 2 iberdomide combinations. So one being the combination of iberdomide with daratumumab and dexamethasone, and the second being the combination of iberdomide, bortezomib, and dexamethasone. And so I think important to say that, again, both of these arms had very, very heavily pretreated patients, so median of 4 prior lines in the Iber-Dara arm and 6 prior lines in the Iber-Velcade arm. We did see quite good response rates, again, in this very heavily pretreated population, so 42% in the Iber-Dara arm, and actually 60% in the Iber-Velcade arm. And these are small numbers, and so we're, you know, early days. I was intrigued by the fact that that signal seemed to be a bit more potent for the Iber-Velcade combination, despite the patients being more heavily pretreated than the Iber-Dara group. The safety profile looked reasonable, and so I'm excited to see these combinations move forward. I think important to mention, there is of course, plans for multiple clinical trials looking at iberdomide combinations in the next couple of years. We in the CMRG are planning a combination iberdomide, bortezomib, dexamethasone study in newly diagnosed transplant ineligible patients. We're using sort of an Iber-VD light protocol in this patient population using weekly subcutaneous Velcade. And so I'm excited to see this study, looking forward to moving that trial forward as well.
I think this may be a good option for newly diagnosed patients that are not transplant eligible, particularly since we don't have funding yet for lenalidomide, dexamethasone, and daratumumab first line for these patients. So we do anticipate this CMRG trial with the oral agent iberdomide, which is very similar in how it's administered to lenalidomide and pomalidomide, and see what sort of benefit we can get. One of the other molecules that has been studied in various trials in Canada, in particular, is selinexor. And Dr. Darrell White has led and participated a number of studies with this, and could you give us an update of abstract number 726 with selinexor?
Sure. So this was one of the arms of the STOMP trial that has been run at many Canadian centers, and this is a platform study essentially that combines selinexor with various known myeloma backbones. And we have had a lot of uptake of these studies in Canada. And additionally, of course, we've enrolled on the Boston trial, which was the randomized phase 3 of selinexor Velcade-dex versus Velcade-dex. So this particular trial is combining selinexor with pomalidomide dex in relapse refractory patients. Selinexor itself is a first-in-class oral selective inhibitor of exportin-1, which is a nuclear export protein. And by blocking this protein, the tumor cell is not able to move tumor suppressor proteins from nucleus to cytoplasm, thus leading to tumor cell apoptosis. The drug had been studied in the US on the STORM trial with dexamethasone, and by itself with dexamethasone, there's about a 26% response rate, which led to its FDA approval. So this particular trial was the first combining with pomalidomide and dexamethasone. It is dose finding and then dose expansion with the recognition of the phase 2 dose. So we did begin with, in this case, a relatively higher dose of selinexor in terms of being combined with pomalidomide and dexamethasone at least. And with subsequent groups of patients, the dose was brought down such that the recommended phase 2 dose was found to be selinexor 60 milligrams once a week, along with standard dosing pomalidomide and dexamethasone. And that's a little less than half of the amount of selinexor that was used on the STORM trial, for example. The population of patients enrolled, so about 65 patients overall. 20 of the patients were enrolled at the phase 2 dose. The patients were heavily pretreated with 3 prior lines of treatment. 100% had seen lenalidomide, and close to 80% were lenalidomide refractory. Hematologic toxicity was common, about 50% neutropenia. Interestingly, the GI toxicity that's so dominant with the higher dose of selinexor wasn't a problem so much in terms of the higher grades of toxicities. So very little grade 3, 4, but grade 1, 2 still quite common, with GI toxicity being nausea, vomiting, diarrhea, and weight loss. In terms of effect, overall the response rate's about 50%, and at the phase 2 dose, response rate's about 60%. Progression-free survival is, the trial is relatively immature at this point, but for the overall population, it's in the range of a year and not reached for the phase 2 dose. So overall, I think the data is promising, that the combination's tolerable at these doses, and the hope is to carry on to phase 3 dosing.
Thank you. I would add that selinexor is available through a special access program, again, through Health Canada. And since our drug approval and certainly funding processes are slower than in the US, we do rely on these programs to try to provide our patients with the newest drugs available. Ajai, do you have any comments about selinexor? Because it is on the market in the US, and you've had the opportunity probably to consider its use more readily than we have.
Yeah, I think it's important to distinguish the population. STORM, very heavily treated. Many of these patients could have gone to hospice. They had a median of 22% increase in their paraprotein between the day of screening and start, so 12 days. And so when you have that population, you need 80 milligrams twice weekly with dexamethasone. And the point of that study was really to demonstrate single agent efficacy with dexamethasone. But now that it's approved, and that's what I'm looking for with the belamaf too, there's a difference between getting these accelerated approval and then what you actually want to do with your patient, because myeloma is too genomically complicated to depend on just 2 drugs. Study after study is showing triplets are better. And the FDA even said, why do we keep doing doublet comparisons? It's like we're comparing against this archaic backbone. But so Fortunately, the STOMP study, as has been beautifully presented, you have a choice. You can, if you have somebody who has cytopenias, you can combine with CD38 or PI. If you want an oral regimen and you have good blood counts, use an IMiD, right? So it's nice to have that flexibility. And I think more drugs need to do these kind of backbone studies so that you can tailor the therapy to each patient, given their, AE profiles.
Thank you. And now that we have Dr. Song to join us, Kevin, would you introduce yourself? And then we're going to ask you to present 2 more abstracts that loop back, first of all, to allogeneic CAR T cells, and then to a carfilzomib-based regimen.
Hi, Donna. I'm Dr. Kevin Song. I'm a hematologist at the Vancouver General Hospital with a specific interest in multiple myeloma. This was a very interesting ASH year, and I'll be presenting the 2 abstracts that you had mentioned. The first is an allogeneic first-in-human study of the anti-BCMA ALLO-715 and the anti-CD52 ALLO-647 in relapse refractory multiple myeloma. I thought this abstract was extremely interesting because, as you all know, and as was discussed with the rest of the colleagues here, there's a lot of activity that's happening in the anti-BCMA space, and particularly CAR T cells are becoming very advanced, autologous CAR T cells in particular. But autologous CAR T cells have their own problems with, having to collect the T cells and manufacture them, and then eventually giving it to the patient. So it does take a significant amount of time for the patients to get the CAR T cells. And there's obviously particular issues with the person's immune system, given that they're pretty much heavily treated at this time. So there's a particular attraction with allogeneic CAR T cells, and this was a study that was presented by the group in New York in combination with the group in Wisconsin. And when they presented this abstract, they updated their results, and essentially they had these CAR T cells that were developed such that the T cell receptor was disrupted, as was CD52, and this was because of the concern of graft versus host disease, which can develop when you get allogeneic CAR T cells. They managed to enroll about 35 patients, of which they were able to evaluate 31 of these patients. Median age was 65. 50% actually had high-risk cytogenetics, and 94% of the patients were penta-exposed. The median follow-up was relatively short at about 3.2 months, but importantly, they were able to collect some important data. Particularly regarding safety, about 45% of patients had grade 1 to 2 CRS, but only about just under 20% required tocilizumab and or steroids. Infections were found in about 42%, but they were not necessarily very severe infections. And no patients had graft versus host disease, which is of course very reassuring. Importantly, when they were assessing the efficacy, they had about 10 patients that were given the highest dose levels that they were giving. And of these 10 patients, about 60% or 6 out of 10 had actually a response, of which 4 out of these 10 had a very good partial response. And the response was relatively quick. It was within about 16 days. And the longest response that they have is a patient who's really out about 6 months. And the presenters mentioned that this person was actually out working because the patient is feeling so well at this time. And so this study was extremely impressive in terms of how well it was tolerated, the degree of toxicity that they had. And they demonstrated that the higher doses, the efficacy was relatively quite high. So hopefully they'll be able to embark on enrolling more patients and have more clearer data. And this may be a very viable option for patients who require some type of anti-BCMA therapy.
Thank you. I think one of the obvious points, and we've used the term off the shelf, I think, in some of these discussions, one of the pros of the BiTEs is it's off the shelf. You can pick it up and administer it. And one of the challenges with the autologous CAR T cells is there is quite a significant lag, weeks after you collect, when the cells are processed. And it's been difficult to compress that any shorter than about a month. And a lot can happen to your patient in a month to the point that they can't get their CAR T cells if their myeloma progresses too quickly. But the allogeneic CAR T cells are off the shelf. They don't come from the patient, they come from somewhere else. You can order them and get them in. You can standardize the product. You don't have to sort of be stuck with what you get from an immunocompromised patient. And the fact that they proof of principle that you can do this without GVHD, I think really makes some of the limitations of autologous CAR T cells less important. So I think I echo that. This is very exciting. We do have a trial ongoing of a different allogeneic CAR T cell platform that's open at our center, and I think there may be other Canadian centers that will be embarking on this kind of study over the next little while. Dr. Chari, have you had experience with any allogeneic CAR T cells yet?
Just have this study opened very recently. So, but I think you hit the nail on the head. You know, this is going to be the key question. If we had to do CAR T versus BiTE study, the intention to treat would immediately favor BiTE over the auto CAR Ts because of all the issues we've been talking about. But here you could really do a head-to-head study. And I think that even if the efficacy is a little bit lower than autologous CAR, the PFS benefit that you can be sure is coming from the CAR as opposed to bridging chemo and patient selection, I think is a real winner. And to me, if this data is mature, it's the allo CARs to lose at this point in terms of comparing to auto CAR. And not to mention, you know, being able to deliver it, you know, cost considerations, manufacturing, and also I think the safety looks very compelling, you know, primarily grade 1, 2, no severe neurotox. I think it's really very, very exciting.
I should add that this is abstract number 129 for those in the audience that are tracking these studies. And Dr. Song, I'll ask you to move on and discuss another interesting abstract because it mirrors a CMRG study that was done and reported well before this one. It uses agents that have been available to us, including carfilzomib. So this is abstract 415. If you would give us a summary of that and your thoughts, please.
Absolutely. This was also a very interesting abstract. It's not as novel as the one that I just presented, but perhaps it's more practical in terms of, you know, something that we can do tomorrow. So this abstract 415 is titled a randomized phase 2 study of weekly carfilzomib 70 milligrams per meter squared and dexamethasone plus minus cyclophosphamide in relapse refractory multiple myeloma. And this was done by the Spanish group. It's a rather large size randomized phase 2 study with just under 200 patients who were enrolled. And as mentioned, all of them got carfilzomib at 70 milligrams per meter squared weekly with dexamethasone. And half the patients were also randomized to get cyclophosphamide at 300 milligrams per meter squared, but they were given this drug intravenously. And so what was really important was that you know, patients may have received prior proteasome inhibitors, but they were not refractory to proteasome inhibitors. The median age of the enrolled patients was about 70. About 25% of the patients had high-risk cytogenetics. And what their data showed was that, first of all, virtually all patients had received PI. 92% had received bortezomib prior to enrollment. And 66% actually had IMiDs as well too. And just a small number, 4% had anti-CD38 antibodies. And importantly, 35% of patients were actually refractory to IMiDs. So what they noticed was that the PFS was actually improved in the arm that did receive cyclophosphamide at about 20.7 months versus 15.2 months. So a 5-month improvement, but the p-value was not significant, it was 0.24. When they did multiple sub-analysis, what was most interesting were that the patients who were IMiD exposed, as well as the patients who were IMiD refractory, they seemed to benefit the most. For those who were LEN refractory, the benefit was such that their PFS was 26.2 months versus 9.3 months, and that was significant. And what they remarked on was that this combination was relatively well tolerated. There was slightly higher neutropenia, but otherwise there was really no other signals of concern. Importantly, I think, as you had mentioned, we have done and presented at previous ASH the CMRG study, or back then it was the MCRN 003 study, which looked at carfilzomib at the same dose weekly, 70 milligrams per meter squared, in combination with oral cyclophosphamide. And I believe the numbers are pretty similar to what we found here.
Yes, just to highlight a couple of points, we used oral rather than intravenous cyclophosphamide as was administered in the Spanish study, so that's another cost savings. Also, we limited the cyclophosphamide in our Canadian trial to just 1 year, where I believe the current study continued it until progression. And there are concerns with very long-term alkylating agent treatment in terms of potential marrow damage. So I think, again, we were able to produce thoughtful results, and one could even perhaps go out on a limb and say the Spanish study confirmed our findings, but Canadians are by definition modest, so maybe we won't say that exactly.
I would just add, I completely agree, but I think this kind of mimics the other study and the importance of cyclophosphamide in lenalidomide refractory patients, whether you're using it with POM, DARA, or with carfilzomib. And I think, to me, when you zoom out a little bit, there's kind of 4 dance partners in the room. And if at first relapse, it's CD38 with either POM or cyclophosphamide, then the other dance partners go with the cyclophosphamide is available. So I think CD30 with POM or CARF, and then whoever's left goes with Cytoxan. So it's nice to have these kind of options until studies are more available, clinical trial options that are getting to approvals.
Well, I think we've all been interested in Canada about, without the scientific underpinnings yet, of using cyclophosphamide as a cytotoxic and as an immune modulating agent. It clearly has immuno effector type activity in terms of Treg cells and a variety of other activities that have not been well studied. But the Laurentian trial, the 004 trial that was presented earlier with the quadruplet, does have biologic correlatives. So maybe we'll get some insights about the effect of cyclophosphamide. But it is a way to enhance other drugs at, as Dr. McCurdy said, almost a cost neutral. And our funders don't seem to get too upset if we sneak it in. Before we move into the section on stem cell transplantation as part of first-line therapy, I'd like to ask Dr. McCurdy to provide a summary of some really important results on the management of newly diagnosed light chain amyloidosis. This is abstract number 552 that evaluates CyBorD with that agent, that regimen, with the addition of daratumumab.
Thanks, Donna. So this is a really exciting abstract, I must say, comparing in a large randomized trial, one of our Canadian standards for newly diagnosed AL amyloid patients, CyBorD, compared to the combination of daratumumab plus CyBorD. And I think really impressive results. So the response rates, hematologic response rates, were over 90%. And really across the board, when you look at every outcome from this study with respect to hematologic response, organ response, and deterioration of function of the major organs, in particular cardiac and renal, the study favored daratumumab in addition to CyBorD. And, you know, this was a very well tolerated regimen. I think it's very relevant for our practice. As we know, we don't currently have any approved or reimbursed options, treatment options for patients with newly diagnosed AL amyloidosis. And so we're really hoping that this could be a new standard of care in Canada. So I'm very excited about this. It will be a fantastic addition for both patients and treaters of this very difficult illness.
Thank you. And I think the use of subcutaneous daratumumab makes this very user-friendly for a population that may have issues with fluid retention. So thank you. Now moving on to the topic of newly diagnosed patients, beginning with trials that look at the potential role of autologous stem cell transplantation. And we had 2 abstracts or presentations this year that provided more information on the large randomized EMN02 HOVON 95 trials. And I'd like to ask Dr. Leblanc to present the first abstract, which is number 142.
Thank you, Donna. So the EMN02 HOVON 95 phase 3 trials in newly diagnosed myeloma patients include 2 randomization, and this session is about the intensification randomization comparing autologous stem cell transplant to no transplant using VMP. And the study has been already published, but it was with a shorter follow-up with an improvement in PFS, but no change in overall survival. Now with a significantly longer follow-up, they look at different endpoints, including overall survival. And specifically, the 75 months overall survival was 69% for the transplant group versus 63% for the VMP group without transplant. And in addition, patients receiving delayed autologous stem cell transplant, meaning that they were randomized to VMP intensification and at time of relapse received autologous stem cell transplant. Even in this group, they had a shorter PFS2 and overall survival compared to patients receiving upfront autologous stem cell transplant. So the studies certainly support the use of upfront autologous stem cell transplant in newly diagnosed patients who are fit enough to high-dose melphalan. And what is important to note is that the significance of these clinical trials support to not change the treatment decision made in newly diagnosed myeloma patients, because it directly reaffirms what we are currently doing present in Canada.
Thank you. And I'd like to ask Dr. Stakiw to present another aspect of this study, which isolated the effect of consolidation, which was given to half of the patients. This is summarized in abstract 550.
Thanks, Donna. So yeah, just over 870 patients were randomized in the second part of the study to VRD consolidation for 2 cycles, followed by lenalidomide maintenance, or just lenalidomide maintenance alone. And again, progression-free survival seemed to benefit the consolidation arm in almost every subgroup that was studied. Certainly in the standard risk and in those who obtained a complete response after the transplant, they did better, but across all of the subgroups, progression-free survival was sustained in those who got the VRD consolidation. And so in full disclosure, we've been doing this in Saskatchewan now for 18 months to 2 years. I'm not sure how we got it funded. The 87% of people who went into the VRD consolidation completed it. And so I think we were talking about that earlier. Was it 87 or 97? Sorry. But a majority of people who went into VRD consolidation completed it. I find it a little bit difficult to give the VRD consolidation for people who've had trouble getting through the transplant and so quickly after transplant. But I think that this, as opposed to other trials, could be a new standard of care in Canada.
So you are lucky in Saskatchewan
yes.
to have been able to secure consolidation. We have not in Ontario, and I think most provinces have had challenges. Ajai, do you use consolidation in a more permissive funding environment?
Well, I think what's key about the study was they didn't have lenalidomide up front, right? And so it really it's difficult to apply to the US population where we have extensive len use, but I think it is philosophically a very interesting topic. You know, I just today had a patient we were talking about post-transplant. You know, it's like a boxing match, and if your opponent is down, that extra final knockout punch seems surprisingly, you know, this brief consolidation translating to PFS, it's a little bit mechanistically surprising, but the data are what they are, and I think it speaks to the importance. And I think the other thing is the fact that, as Julie was mentioning, even CR patients benefited. You know, sometimes we think we know who to do what in, but it's not clear, right? Sometimes in both in the Stamina study and in the first study, discontinuing len didn't, they actually, the people who do better, right? So I think we need more risk-adapted therapy going forward. But I do consolidation if they haven't achieved remission, not on the basis of great prospective data.
But I think your point's well taken, and it is hard to sort out because what the EMN trial used, and we discussed this earlier, was a cyclophosphamide bortezomib dex induction, which is similar to the CyBorD we use here. So we don't introduce in Canada routinely lenalidomide until maintenance, but our real world data of CyBorD transplant lenalidomide maintenance in Canada in over 700 patients yielded a progression-free survival of 58 months, which I think is competitive with other platforms in the previous era. But we'll hear an update of the IFM trial next, but then we'll move in to adding some of the newer agents to the stem cell transplant sequence that may take us a step further up in our outcomes. So I will then ask Dr. Song to provide information on abstract 143, which does look at one of the other major randomized trials comparing upfront transplant to delayed transplant using RVD as induction.
So this is a trial that we were all quite familiar with. It was published initially in the New England Journal of Medicine about 3 to 4 years ago. Essentially 2-arm trial. One arm gets 8 cycles of RVD, the other one gets 3 followed by an auto transplant, then 2 cycles of consolidation, then they get lenalidomide maintenance. Essentially, this looked at what happens at relapse, and the great majority of patients who relapsed on the RVD times 8 group proceeded onwards to get autologous stem cell transplant. So what essentially they got was delayed transplant, whereas the majority of patients in the other arm who did get the transplants got further therapy. Interestingly enough, the treatment that most of them got was pomalidomide because carfilzomib and daratumumab were not readily available in France around that time. Just to get to the punchline of the study, essentially, the overall survivals were very similar in the 2 arms. They were about 60% at 8 years. The PFS2 was very similar as well too, and essentially they said that patients can receive a delayed transplant, and this does not seem to compromise their overall survival. And I think this is obviously slightly different than what was just presented with the EMN study, and maybe goes back to what Dr. Chari had mentioned regarding the use of lenalidomide in this study compared to the other study.
I think the other point that makes it, again, very hard to sort out what is doing what is that the IFM study truncated lenalidomide maintenance at 1 year. So patients were not progressing on lenalidomide in general. So I think that to me makes it hard to compare with other trials that have looked at delayed versus early transplant when lenalidomide is given like we now give it most of the time, which is until disease progression. One thing that I really feel it is important to have data regarding to advise our patients, very hard to dig out is what is your PFS with a delayed transplant? Because we all have discussed that there is a recovery time from a stem cell transplant. People are tired, they're not up to speed for several weeks or even months. And if you don't get a lot of bang for your buck in terms of PFS with your stem cell transplant, I think patients need to know that. And the EMN study did not indicate, they only have given PFS2, but it doesn't look like you get more than a year with a delayed transplant. And even though the survival is the same in the IFM trial, I really think that's an important piece of information to help our patients decide if they wanted to go to delayed transplant, which I think I personally don't feel is the right way to go, but I respect other people's opinion.
I would agree with you, Donna. That's how I felt about this abstract, really, that it reinforced that you get a bigger bang for the buck in terms of PFS with the first transplant. And although you can get something out of the second, a delayed transplant, really you get more of a remission with the first transplant. So I would recommend that for my patients still.
And if I can add, eligibility criteria for transplant are harder to meet compared to chemotherapy alone. So if a patient is eligible at first-line treatment, why to wait? The attrition that we see for second, third, fourth line of treatment, there is no goal of that. So if a patient is eligible at first line, I really think with the data we have, with the tools, with the access of medication we have in Canada, transplant is still appropriate in first line treatment.
Thank you. So I think now we'll move to maybe think of it as enhancing this, this stem cell transplant. And I will ask Dr. White if he will share with us updated information on the Italian Forte trial, which is abstract 141.
Thanks, Donna. So I agree with previous comments as well, and certainly this Forte trial is definitely sort of stepping it up. So this is Transplant Plus, and I must say this is one of my favorite trials to follow. So you know, there's I think as a trialist, it's very nicely designed. There's a lot of questions that this is going to answer in time, and we've seen parts of it before in this study, in this meeting. We're seeing more of it, but it is complicated. So this is an Italian intergroup trial with 3 arms, 2 randomizations. The 3 arms are carfilzomib, len, dex, for a total of 12 cycles with no transplant, carfilzomib, len, dex for 4 cycles as induction, then an auto transplant, and then another 4 cycles as consolidation. And the third arm is carfilzomib cyclophosphamide dex in the same manner. So 4 cycles induction, transplant, 4 cycles consolidation. And then the second randomization, which is being reported on here, is randomizing the patients that make it through to that point to either lenalidomide maintenance or lenalidomide and carfilzomib maintenance. And what's the primary endpoint is progression-free survival at the 2 randomization points. And essentially what's been shown in the past is a better response rate for the carfilzomib, the 2 carfilzomib arms, actually better than KCD. And what's being shown now is that progression-free survival is actually best for the KRD with transplant, better than both the KCD with transplant and the KRD continuous. And then beyond the second randomization, the carfilzomib len maintenance is superior to the len maintenance alone. And there's a couple of additional caveats here. So there is MRD data that is very interesting, and they're reporting on 1-year sustained MRD rates, which are also better with the most intense treatment. So they're attaining about a 68% 1-year MRD sustained rate. The PFS overall is quite high. So 3-year PFS in the KRD transplant group was 78%. And then moving on to the maintenance, they're seeing a PFS at 30 months in the range of 80% with carfilzomib len. And in both portions of the trial, they're seeing benefit extend across subgroups, including high risk. And the high risk is not yet equivalent to the standard risk, but it's approaching. Overall survival is immature, and that'll come in time. Safety is, it appears tolerable, but there is more toxicity certainly when carfilzomib is used in an intense fashion. Discontinuation for all arms was actually reasonable in the range of 9 or 10%. And I think that's the information so far. So I think that the takeaways are that this is another trial confirming transplant is a benefit. It also says that consolidation is probably important, And it tells us that we can probably do better than lenalidomide in terms of maintenance treatment.
I think we're hearing those themes over and over again. One aspect that I find reassuring is that for our high-risk patients in Canada, a number of us are empirically using lenalidomide, which is widely funded, and PI, not carfilzomib, but either bortezomib or often ixazomib. And this is very supportive of that. And we will use our database when some of this experience is more mature to actually look and see what we've done. But I think it's always reassuring when what we think will help patients, at least in concept, has been demonstrated to do that. So the next trial, everyone's been watching with a great deal of anticipation. Dr. Chari, this is abstract 549, which is nicknamed the Griffin trial, and adds a monoclonal antibody from the beginning in a transplant-eligible population.
Yeah, so in full disclosure, I'm a co-author on this. So, but I think we know that Cassiopeia, DARA, VTD in Europe, compared to without the DARA in a transplant-eligible population, resulted in not only improved depth of response, but PFS. And so the question is, and that was only with 18-month follow-up here, what would be the outcome? So this has already been published in Blood, the first iteration that showed better depth of stringent CR, and this is with additional follow-up of 27 months. And we see that additional deepening of this depth of response, stringent CR, 63% in the DARA VRD versus 47% in the RVD. We also saw deeper MRD, and not just one time point, but sustained MRD was better with the addition of DARA, 28% versus 2.9%. PFS, I think because RVD is we've been saying RVD is a pretty good backbone. So with 27 months, we may not see much. And that's actually what we saw, that the PFS was at 2 years was 94.5% versus 90.8%. We do see some additional neutropenia with frontline DARA. But to me, what I take home message for this is, what are our induction choices right now for the US? It's RVD, DARA RVD, KRD, and then DARA VTD based on Cassiopeia, although we don't use it. But I think the endurance study showed us that KRD and KCD are really there's not a superiority. And the Forte, which we just heard about, is very important because even what we don't pay attention to is, I have a slide summarizing this, how many cycles of chemotherapy are we talking about for each of these regimens, right? So the Forte, 12 cycles, how many of us give twice weekly carfilzomib for a year? And talk about physician fatigue, patient fatigue, healthcare systems fatigue from a cost point of view. So if KRD for a year could not displace transplant, it just tells you that transplant's not going away anytime soon. Now, when we look at these other regimens, Griffin is relatively short. It's 4.5 months of DARA in terms of when we look at induction and consolidation, which is not that much. And so I think my closing statement on this would be, are you an early adopter or not? And some people want that. I think to ask for OS in a newly diagnosed study will be retired practically. PFS is a reasonable surrogate, but even that is going to take a long time, given how we've seen with the IFM data. So the question is, do you believe in MRD? And the only time I believe in MRD is in exactly this setting, which is in a clinical trial in a rigorously done fashion. So to me, this is an early readout, and I've adopted RVD. And I would just say, though, I've actually used weekly bortezomib because, again, I think twice weekly is challenging. So from a cost and practicality point of view, I like the convenience of weekly RVD, and then the additional CD38, I think, is giving me that deeper response. But I think this was really interesting data, and I'm an early adopter. But I'm curious what you guys think.
To dream to be able to get DARA up for transplant.
Yeah.
So back to our CMRG goals and trying to get these drugs into our Canadian patients as soon as possible. We will have a trial early next year that will add the other CD38 antibody, isatuximab, to our transplant backbone, which is CyBorD stem cell transplant, and then 2 years of isatuximab, similar to the daratumumab given for 2 years after the RVD. So I think this trial will likely accrue very quickly and hopefully buy us some time until we can get more broad monoclonal antibody support.
Bring up a very important study design difference between Cassiopeia and Griffin. This is DARA continuously in the DARA arm for the whole 2 years. Cassiopeia, after the induction and consolidation, you could get randomized to placebo. So it's a different question and of DARA upfront versus DARA in maintenance. Here it's DARA throughout, as you're doing with ESA, and I think it'll be an important study. So kudos to getting CD38 for your patients.
I think another thing, thinking optimistically, in Canada, bortezomib is off patent, and lenalidomide will go off patent very soon. So I think if we are combining a quadruplet, RVD plus DARA, and if 2 of those drugs have come down in cost, I'd like to think we have some point to negotiate with our funders as these trials mature and offer patients state-of-the-art outside of a clinical trial.
They did, there was just Health Canada approval for DARA VTD based on Cassiopeia. So I don't know what that's going to do in our landscape. I don't imagine there's going to be major uptake of DVTD, but if Revlimid comes off patent, we may be able to work some magic there. So we'll see.
Yeah.
How come Canada didn't adopt the IFM, which showed that VTD was better than VCD? How come you guys really like your cyclophosphamide, huh?
Well, we don't have RVD funded, but the VCD given in the French trial was quite different from the way we give it. They give high dose cyclophosphamide, which my personal bias is not as effective as low dose, where I think you may get more of the immunomodulatory effect. Because our results with CyBorD induction after 4 cycles are consistently over 90%. Now, we don't have detailed MRD negativity rate. It's not going to be as good. But if you can catch up with using lenalidomide for maintenance, we do better than one would think from that French experience. But I think there are nuances in how you give VCD or CyBorD. And of course, we use weekly bortezomib, and we give 1.5 milligrams per meter squared. So we don't run into the neurotoxicity where we have to discontinue our dose reduce that can be seen with the twice weekly. And maybe there's something to giving a little bit higher dose once a week. So I think we're all very aware of the French data, but our regimen is different, and we have 2 analyses, one locally from Princess Margaret and our national trial in a large number of patients that I alluded to earlier that show it is a workhorse for us. And that's not just sour grapes.
Well, based on that, your challenge to Forte might be then their KCD was the not Canadian KCD, and you could do one better.
The data's the data, so I don't think we'll turn down a PI lenalidomide combination up front, but I think we have looked at our data in a large population, and we hit pretty good benchmarks. And just to be very provocative, if you look at the IFM data, which were trial patients and lenalidomide for 1 year, their PFS in the transplant arm was 50 months. Our PFS, not trial patients, CyBorD transplant lenalidomide into progression, was 58 months in the real world. So, you know, I don't think we have to be apologetic, but we can do better, and we can look forward and try to get the new drugs in as quickly as possible. But we're proud to be.
So being Canadian is independently a favorable prognostic factor. I see a nodding across the board. All right.
Or perhaps we're less modest than we initially hear.
So thank you very much for that discussion. In the last two abstracts, we're going to change tacks again, and Dr. White will present the phase 3 tourmaline trial, which is abstract 551, and then we'll finish with Dr. Song with abstract 548, which looks at a completely different setting, which is high-risk smoldering myeloma. So I'll ask Darrell to take it away with the tourmaline trial.
Thanks, Donna. So this was the tourmaline 2 trial, which was the frontline trial many of us participated in. And you know, I'll sort of state up front that I think it's a very nicely done trial. It's placebo-controlled, which we haven't seen that often in the large phase 3 trials. Looking at frontline treatment for newly diagnosed transplant ineligible patients, large trial with 705 patients, and unfortunately it's a negative trial as well. So it did not meet its PFS endpoint you know, of IRD versus placebo RD. However, there is a numeric advantage that's over a year in median progression-free survival in the two arms. I think about 13 and a half months. And it's, you know, for what it's worth, it does come close to being statistically significant, which probably will be talked about, but I don't think we'll get it very far with the regulators. There is a bit of nuance in the trial, though, that's interesting. So this did include, or did define high risk as including 1Q abnormalities. So the proportion of high-risk patients in this trial is higher than some of the others, ranging up to about 40%. And you remember from the tourmaline 1 trial, the subset of high-risk patients appeared to do better with the combination of IRD. And in this trial as well, the subset of high-risk patients appears to do better with that combination, albeit it's a slightly different definition of high risk than some of the other trials. It's tolerable and it's more convenient than KRD or VRD, but again, it doesn't stand the test of statistically significant PFS improvement.
I think that many of us participated in this trial, and I don't want to speak for everyone, but I think having an oral proteasome inhibitor does scratch an itch for certain patients. I see Julie nodding, particularly those that don't live in a large urban area. And we have been very fortunate to have a compassionate program to access ixazomib without funding, despite Health Canada approval in the relapse setting. So we've been able to offer these patients the advantages of an oral PI. I don't know if anyone else has comments about.
I would just add, I think for two categories, rural patients who have to really drive a long distance, I think this is really an important value add. And the other thing I think is just to keep in mind in the back of our minds, the COVID-19 situation, right? So our paper recently came out in Blood, and if you think about myeloma patients, the Spanish group did show that the mortality rate is 30% versus 20% for non-myeloma age-matched controls, but the people who do worse within myeloma in terms of high risk of mortality are older, high-risk disease, renal failure, and uncontrolled myeloma. And so I think we need to think about our patients differently. So if you have a frail elderly patient, you don't want to compromise on their myeloma control, but you also don't want them to expose them to recurrent virus. So I think it is nice to have all oral options for these patients to keep their disease under control without having to risk their COVID exposure going up.
Good point. And then finally, turning to another topic about even earlier management of myeloma, or attempts to modify the natural history of myeloma by treating it before the actual manifestations of malignancy become apparent. So Kevin, would you give us your take, please, on treatment of smoldering myeloma?
So this was a really interesting abstract presented recently at the ASH meeting. And so they took patients who had high-risk smoldering myeloma based upon the Spanish and the Mayo criteria. And essentially, they gave them 8 cycles of KRD followed by about 24 months of lenalidomide. They enrolled in the study roughly about just over 50 patients. And what was really interesting was that the patients did surprisingly, well, not surprisingly, but they did remarkably well. They tolerated the treatments really well. The follow-up was about 28 months and beyond. The MRD was 70% actually were MRD negative. And for those who are MRD negative, the median MRD CR was about 66 months. And so for 7 years sustained MRD was 40%. Progression to myeloma only happened in about 2 patients. So there was 91% progression-free at 8 years, with 80% biochemical progression-free at 8 years. Overall survival was 100%. So I thought this trial was very interesting because I guess one of the issues is, for patients who have smoldering myeloma who don't have clinical myeloma, is it possible that you could give them a treatment to potentially cure some of these patients? And certainly when you're getting to 8 years and beyond without progressing, it's really, really remarkable. So it's very attractive, I think, compared to some of the other studies, which are also very good, but this one seems to be a bit better than them.
Thoughts from the group?
This is my pet peeve, so maybe I have to get it off my chest. So I'm going to take off my American hat and put on my ex-US regulatory hat. Any phase 2 study in smoldering is a hypothesis-generating study and requires validation by a phase 3 study. And we need to stop looking at patients as photographs and look at movies, because this study, and I was noticing, if I were a reviewer or of any journal with a smoldering myeloma, I would kick it out if they don't tell us what's the median time since diagnosis, right? Because we're supposed to be focusing on those first couple of years in preventing disease. And so I've had patients leave Mount Sinai, well-educated professors who've had smoldering for 4 years, and I said, look, you're not high risk because you haven't progressed. They'll go to an institution down the street and get enrolled in a smoldering study. So is that the patient that we should be treating? So I really think we need to think about, there was a VA study that showed that the typical active myeloma patient has had myeloma had detectable for 10 years prior to the diagnosis. All of this is speaking to lead time bias. You need to think about how long people have had it. You need to think about the risk of therapy, the toxicity, the cost. You have to collect stem cells in these patients, the discontinuation rate, the heart failure rate. These are healthy people. Every toxicity is unacceptable, really. And at the end of the day, I don't care about PFS because we've already seen the impact of len-resistant in frontline myeloma. So now you're taking smoldering people and making them len-resistant. So these are hypothesis generating. I want to see the PFS2 data. I want to see the OS data, which we're never going to get. I know that. But at least let's narrow the field and talk about recently diagnosed smoldering. I guess that was, yeah, this study didn't even present that information, which is, median time since initial diagnosis.
Thank you, Kevin, for leading us into that great discussion. And thanks everyone for their participation and insights in these presentations. So now we will open the forum up for the question period. I'd like to remind our viewers that you can send your questions in the YouTube live chat, and we'll try to answer as many of them as possible. Thank you, everyone. You've heard throughout this presentation that we have a whole generation of patients that are receiving lenalidomide as part of first-line treatment, either as maintenance or with RD for non-transplants, and now are progressing. In Canada, they're usually on treatment. They haven't had a hiatus. We tend to give the drug as long as possible based on the data. So from the different presentations, which have given carfilzomib, Dex, there's also carfilzomib, daratumumab dex, there is pomalidomide, Dara dex. Do we have a new standard, assuming that these regimens will be available to us. I'll see if anyone wants to discuss if we should develop some sort of consensus for second-line therapy first, and then maybe call on some people.
To say a new standard, it might be hard presently because we have many options, but that's what we want to have, many options to be able to individualize treatment for each patient. For example, for IKEMA trial, this is a very important trial for those patients who have not been exposed to anti-CD38 monoclonal antibody in the past. At the time they received carfilzomib, I think the addition of this agent, isatuximab, is certainly appropriate. It increased the response rate, quality of response, duration of response. So we need more and more options to try to individualize treatment to each patient.
I was just going to add, I agree with Richard, and I think Dr. Chari commented on this earlier today. You know, in this Rev refractory population, we at least we've seen even updates from ASH of the Candor trial with DARA and CAR, the IKEMA trial with ISA and CAR, the Apollo study with DARA and POM, and our CMRG study with DARA, Cyclo, and POM. And I think that, you know, I'm guessing that most of us would favor one of those options, depending on what's available in our respective provinces and some of the individual characteristics of the patients. And I think we're all very excited by the quadruplet of our study. But I think it'd be difficult maybe to have a consensus. But I think one thing we could have a consensus on is that our current reimbursed option in this setting is not good enough. The combination of DARA, Velcade, and Dex, which is provincially reimbursed for Rev refractory patients, is not good enough. The response rates and PFS are not good enough in this setting, especially with these other agents that are readily available. And so that would be where I would be advocating for, you know, these other combinations in this Rev refractory population should be considered because the current standard can be improved on.
I think that summarizes nicely, and I think we are glad to have a DARA-based regimen for relapsing patients on lenalidomide. Otherwise, we would not be able to introduce a CD38 antibody as second-line treatment. But the trial that led to approval and funding of the DARA-VD regimen has not given us outcomes for Rev refractory first relapse patients. And so we know it's not as good as the whole group. There are patients who will do very well, but the median is not what we want it to be, as you pointed out. So we're ready to move on beyond that. I think your point that it's premature to pick one regimen, what comes to mind is we need to look at these 4 studies. It's cross-trial comparison, which is a no-no, but we do it. But we need to see how many were Rev exposed and how many were Rev refractory, because that makes a difference. And in some of these studies, many had Rev, but they weren't relapsing on it. We know they're likely to do better with anything. We have variable percentages of high risk and extramedullary disease. We know those factors can determine the outcome in a way that is independent almost from the exact drugs we use. So when we have those types of information, we may see if there is one or another that is preferable. But as Richard pointed out, we want more than one regimen because patients are variable, both in their own characteristics, if they have bad blood counts, if they have cardiac disease, if they have peripheral neuropathy, et cetera, et cetera. But maybe I will ask another question here.
I had just one point before you do, Donna. I agree entirely with the comments made, and you know, I think Arleigh did a very nice job of highlighting the Michael Seabag's presentation of the CMRG trial with Darapom Cyclodex. And I think the right answer still in Canada, given our funding, is for patients to participate in a clinical trial. And we've been fortunate to have the CMRG trials that we've had, as well as industry trials. And, you know, honestly, and if it's a DVD versus a well-designed clinical trial, the right answer is clinical trial.
Could I also make a comment on the thoughts? I think that they're all appropriate, but I do think that we need to have some type of at least a prevailing idea as to what to do next in this refractory setting. And I think it's obvious that one of the things that we have to think about is that at some point or another, the patients have to get an anti-CD38 antibody, whether it's cetuximab or daratumumab. And it seems that the bigger question is what the partner is going to be, whether it's going to be carfilzomib or pomalidomide. I mean, one of the difficulties that we have is that as we march down the treatments that patients have, unfortunately, our patients will eventually relapse with the next line of therapy, and we need to think about what's beyond that. And most clinical trials require patients who have had exposure to daratumumab. And if they don't have exposure to daratumumab at this point, they probably won't, given the current funding environment.
I think it'll be interesting to see how this plays out too. I don't know the exact timeline, but I understand pomalidomide will come off patent likely well before carfilzomib, and that may affect what we have access to. There is a question here that is not exactly related to what we've talked about, but I think there would be an appreciation of our opinion. The question is, was there any new information regarding the treatment of solitary plasmacytoma of bone? Not a soft tissue, which can be radiocurable, but solitary plasmacytoma of bone, which we know carries an eventual risk of the patient will eventually develop myeloma in almost all settings. Maybe we could see what your thoughts are on managing these patients, which do come along several times a year. I can take a stab if no one wants to raise their hand. I think this is an area that has been tried to be studied, but it's very hard to accrue enough patients because you can have a solitary bone plasmacytoma. There was an old study from Princess Margaret that said the period of control or benefit depended on the size of the lesion. And some of the smaller lesions went many years with just radiation therapy without a systemic relapse. I think what's a little more problematic is we have those patients that have 5 to 10% marrow plasma cells and then a bone plasmacytoma somewhere else. So do they have MGUS that is locally transformed to a new lesion? There was an attempted study in the US. We were invited to join in Canada. Darrell may remember this. That was either going to radiate and then give 6 months of oral. Do you remember, Darrell? The oral ixazomib,
Yeah.
Lenalidomide and Dex, or just radiation alone. And both arms got Zometa. But even though I think there were only 70 patients in each arm, which would have really helped, they could never accrue enough patients. What do you do when you see one of these patients?
I was just going to add, I did not went to all the myeloma oral sessions that I know of at the meeting and the posters except for Monday, and I did not come across anything specific with respect to treatment for solitary bone plasmacytoma at the meeting. There was an education session on Sunday, which went over the other plasma cell disorders, so specifically plasmacytomas and plasma cell leukemia. And I believe the standard of care would still be considered radiotherapy with potentially curative doses, assuming minimal marrow involvement. I did not see anything at this meeting that would indicate otherwise.
And when we look at 10, 15 years of follow-up, they do not all progress to myeloma. I mean, compared to a soft tissue solitary plasmacytoma, for which the minority will progress 15–20% over like 15–20 years. The percentage is higher for solitary bone plasmacytoma. However, there are some patients who are cured, and it's a very indolent disease. So it's hard to recommend presently something else than high dose radiation therapy in an intent to cure the patient, despite the fact that they are at risk.
I agree with that. That's exactly what I do as well.
I think we have time for one more question, and the question has to do with the role of consolidation after stem cell transplantation, which is something, except for a few lucky provinces or individual physicians that have been able to finagle this, is not something we have routinely funded. But the EMN02 trial clearly showed, isolated a benefit of VRD consolidation for 2 cycles. Do you think this is something we should push for in our treatment algorithm?
Well, I'm going to say yes, because it already exists in our treatment algorithm. And I think I was happy to finally have some evidence that justified us sliding it in there and the way we've been using it. I mean, I certainly think that the design of the trial was nice, and that it did compare drugs that we use up front. I mean, in Canada, we don't generally use a lenalidomide-based induction therapy unless they're high-risk cytogenetics, so you can get it some other way. Going into a solitary one transplant and then consolidation across all of the subgroups definitely showed a progression-free survival benefit. So I would advocate for the rest of you to think about to getting it on board.
Can I also just add that I agree with Julie, and I'm slightly envious. I was also going to just say that one thing we could maybe consider discussing is that, you know, doing a consolidation with bortezomib when we already have standard of care Revlimid is actually not insanely expensive, and something that we could easily study given the patients currently enrolled in the CMRG database. Either doing a cohort of patients and using retrospective controls, or doing a randomized study doing consolidation, because we have many of the mechanisms in place that we could do this relatively quickly and have some good data. So I think perhaps something to consider.
I think one question I have about this approach is, if we had RVD induction, do we need RVD consolidation? The trial that showed its benefit had something like CyBOR-D, and I think we'd want to see perhaps MRD data. And if we go back to the original, one of the original thoughts of consolidation is maybe you could bypass continuous lenalidomide maintenance by giving 2 cycles of consolidation like you do in AML, and avoid the long-term cost and maybe toxicity of long-term lenalidomide, and that didn't work. We know continuous treatment has to help. So I think those are the idea. Do we need it if we had RVD up front, or have you already gotten all you can get out of RVD?
And to go in your direction, Donna, the STAMINA trial used for most patients an optimal induction therapy with RVD for most patients, and they were not able to show that consolidation with RVD again was beneficial in the context that all patients received a maintenance treatment, obviously. So again, we don't know if there is a role of consolidation if there is no maintenance used, but it's hard to not use a treatment that has been shown to improve overall survival, and to use something in consolidation that would be probably better than the induction we gave. It's a bit curious that we are stuck presently with CyBOR-D in induction treatment, and we would like to use a consolidation better than the induction. I would prefer to optimize my induction treatment.
Yeah. I think we've seen that the field has taken a quantum leap from 5 years ago when we were arguing, should we not do transplants at all? Now, I think the Canadians and the Europeans, Australians, Japanese had followed the hard data, but there was certainly a great deal of consideration, particularly south of the border, that transplants weren't needed. And we now have those trials that have matured. But now we're seeing the integration of these new agents into our older platforms, and we're at a whole nother level of efficacy. And then we don't have time really to talk about the other, the further down the road end of the advances. There was a question about which is better, CAR T-cell or BiTEs. I took the liberty of sidestepping that because obviously we don't know. Both are incredibly exciting. And as we get experience, we may have our biases. One's off the shelf, one's more involved, but we have allogeneic CAR T cells that are off the shelf. We'll work the kinks out. So I think the bottom line is we have a lot of strategies, both for newly diagnosed patients and those patients that we had nothing for even a couple of years ago. So I think I'm getting a message that it's about time to close. I'd like to thank the panelists, and unfortunately, Dr. Chari got called away to a medical emergency in New York, which is part of the trade-off of virtual meetings when you're still working in your own institution. And I'd like to thank the panelists greatly for the time and effort putting together this program, and Ogilvy for walking us through all the steps to put together a virtual program, and certainly the attendees and Sanofi for an unrestricted grant to put this inaugural CMRG ASH summary together for us in 2020. So everyone have a great evening and a great holiday season, and thank you very much.